Hi everybody, my name is Janishka Gonzalez, and today we are talking about Aarskog syndrome, especially its biochemical basis. So, before we talk or delve into what is Aarskog, we want to have an understanding of what is a genetic disorder. So, when we're talking about genetic disorder, we're talking about a health problem caused by one or more abnormalities in the genome. So, it can be caused by a mutation in a single gene, which we called a monogenic disorder, or multiple genes, which we call a polygenic disorder. It can be also referenced as a multifactorial inheritance disorder, and it can be also caused by a chromosomal abnormality, which is the changes in the number or the structure of that entire chromosome. So the mutation responsible can occur in three ways. So it can occur spontaneously before embryonic development. In the medical world, we call this a de novo mutation, or it can be inheritance from two parents who are carriers of a faulty gene. This is referenced as an autosomal recessive inheritance, or from a parent with that disorder. So that is called an autosomal dominant inheritance. So we have three ways that mutation is responsibly occurring. It can be spontaneously before embryonic development, or it can be inherited either recessively or dominantly. So, when a genetic disorder is inherited from one or both parents, that is classified, and that is classified as a hereditary disease. So, some disorders are caused by a mutation on the X chromosome and have X-linked recessive inheritance, which this is going to be defined further in the presentation, and we're going to see why it's called an X-linked inheritance. And we see that we have very few disorders that are inherited on the Y chromosome or mitochondrial DNA, and this is due to their size. So, before we delve into RSCOV, we want to give an overview of what we are seeing and what is the RSCOV syndrome. So, when we're talking about RSCOV, we're talking about a rare genetic disorder that is very distinctive. It's very distinctive, characterized by facial features, skeletal abnormalities, and genital anomalies. So we see a very distinctive facial feature, which is a wide set eyes and a very nose tip, and we have very distinctive skeletal abnormalities, which are mostly seen on the hands. It's very rare, but it has occurred that there are genital anomalies as well. So this presentation is going to delve into that biochemical mechanisms, specifically exploring its genetic basis, its pathway, and its signaling. So let's start by introducing Aarskov. So Aarskov is also referenced as Aarskov-Scott syndrome or facial digitogenital syndrome, and it is an uncommon genetic disorder. So it is a rare genetic disorder that is characterized by a distinct combination of facial features, skeletal defects, and in some cases genital abnormalities. So if you see on the right the picture, you will see that we have that very distinct characterization of wide set eyes as well as digital webbing of the hands. So initially, R. Scrub was described in 1970 by the Norwegian pediatrician Dagfinn Aarskov, who noted a consistent pattern of physical features among several male patients within a single family unit. So the disease arises from mutations in a gene. This gene is called an FGD1 gene, and we're going to be defining that a bit further into the presentation. But these genes encode a protein that is very critical for orchestrating cellular signal pathways that are essential for a normal development in male patients or X chromosome patients. So, before we go into what is X-linked recessive inheritance, or what is that FGD1 gene, sorry, we're going to see the symptoms, those very common symptoms that are associated with Ascroft. And we see that it is facial, skeletal, and in some cases genital common symptoms. So when we see facials, we see those widely spaced eyes, we see a very short nose with a very broad tip, we see anteverted nostrils, and we see a very prominent upper lip. When we're talking about skeletal, we then see that it's very short in stature. We have abnormalities as short fingers and clinodactyl fingers. We see a very joint hypermobility, and we have mild joint and muscle problems. So, in some cases, individuals with Arcot syndromes may present undescended testicles, and these are distinctive features in the genital area regarding Ascot syndrome. So, now that we know how Ascot syndrome presents, let's talk about what is X-linked recessive inheritance. So when we talk about X-linked recessive inheritance, we're talking about a genetic conditions that are associated with mutations in genes on the X chromosome. So, our Scott syndrome follows an X-linked recessive inheritance pattern, and this means that the FGD1 gene is located on the X chromosome, and males have only one X chromosome because they have X and Y chromosomes. So that's one X chromosome, while females have two X chromosomes, which is XX. So males have one XY and females have two XX. So if a male inherits a copy of the FGD1 gene from his mother, who carries the mutation from one of her X chromosomes, he would likely develop Aarskog syndrome because he has only one X chromosome. Meanwhile, when we see females with one copy of the mutated genes, they are typically known as carriers and may not show symptoms as they have another X chromosome that is normal. So, male will likely develop Aarskot syndrome because they have just one X chromosome, but females are mostly carriers of that disease. So now let's go into the FGD1 gene. So FGD1 protein is also known as facial genital dysplasia 1 protein, and this is a member of the DBL family of guanine nucleotide exchange factors, also referenced as GEFs, and they regulate the activation of the Rho GTA passage. So the genetic basis of Aarskog syndrome lies primarily in the mutation with the FGD1 gene, and that one is located on the short arm of the X chromosome. So if we see the picture in the screen, we see on the right, on the bottom, that short arm of the X chromosome that is known as XP11.21. So FGD1 consists of 18 exons encoding FGD1 protein of 961 amino acids. So the FGD1 protein contains five important domains, including a putative CRC homology tree binding region, two plechrins homology domain, a DBL homology domain, and a zinc finger domain. So it is a Rho RHO, RAC RAC, guanine nucleotide exchange factor GEF, and that can regulate many physiological functions such as cytoskeleton remodeling, osteoblast differentiation, extracellular matrix remodeling, and cell trafficking. And you're going to see um, how it regulates or the process of regulation regarding those physiological functions. So now let's go with how FGD1 signals and its function. So it's important to know again that Ascroft disease is a very rare condition. So this is going to present a possible general mechanism for how FGD1 activates. So it's not certain. Most literature and most studies say that it is a possible mechanism for. So FGD1 is a very soluble protein, and that protein localizes to the subcortical actin cytoskeleton and Golgi membranes. So we have a very soluble protein localized on the subcortical cytoskeleton and membranes, and it translocates to the plasma membrane in response to extracellular stimuli. So, such this extracellular stimuli can be epidermal growth factor in epithelial cells or TGFB in endothelial cells. So, during TGFB signaling, it occurs a binding of the cytokine to its receptor um, to regulate the CRC family kinase, which subsequently phosphorylates FGD1 at tyrosine residue. Now, again, this represents a possible general mechanism for FGD1 activation. So, the activation of FGD1 leads to GDP and GDP exchange on CDC42. So, this in turn mediates a number of cellular events. So, some of these events include, again, as I said before, a cytoskeleton remodeling, and this is through the activation of J and K, which leads to cell migration. It can also include the activation of ERK1-2 and subsequent focal adhesion kinase activation, which leads to metastasis or the activation of ERK1 and 2, or MLK3 and P38, which leads to osteoblast differentiation. So we see that we have a few events, which are cytoskeleton remodeling, we have osteoblast differentiation, and we also have the cell migration. So FGD1 can also regulate ECM remodeling, which is that can happen through the formation of invadopedia in tumor cells or podosums in endothelial cells. So finally, FGD1 controls the export of cargo proteins from the Golgi complex through CDC42 activation, which can be seen in the diagram that I have on the right. So we see that control, that FGD1 control exporting those cargo proteins from the Golgi complex through CDC42 activation. And the FGD1 SH3, which SH3 refers to that SR, that putative SRC homology 3 binding region. um, so it binds directly to that SH3 domain of MABP1 or cortactin. And FGD1 cortactin binding promotes that CDC42 independent acting assembly by the ARP2 and ARP3 complex. So we have a very phosphorylation at serine 205 by glycogen synthetase kinase 3B, which is also referenced as GSK3B, that mediates the degradation of GD1 at the proteasome. So I know this was a very big and very complicated possible general mechanism for how FGD1 activates, but again, it's only a possible mechanism of how Aarskog actually works. So now let's talk about the drug development of Aarskog. So we have a very rare genetic disorder, and this is a challenge to the medical field because there aren't a lot known about Aarskog, and most recent advances in genomic technologies use what we call as iPSC, so induced pluripotent stem cells, and this represents a groundbreaking avenue in Aarskog syndrome research, offering multifaceted applications that range from disease modeling to potential therapeutic development. So most patients with Aarskog suffer a lot of psychological as well as um, pain regarding their syndrome. But Through the reprogramming of adult somatic cells, such as skin fibroblasts or blood cells, we have those induced pluripotent stem cells, iPSCs, that are generated, which they acquired pluripotency akin to embryonic stem cells. In the context of ARCA syndromes, iPSCs technology represents several opportunities. So, firstly, iPSCs derives from effective individuals that serve as invaluable disease models, and they enable researchers to recreate the syndrome phenotype in vitro. So, remember, in vitro, we have those um, cultures in the lab. I'm sorry, I spaced out. So when we talk about in vitro, we're talking about lab research. So by differentiating these iPSCs into relevant cell types affected by Aarskog syndrome, such as osteoblasts or chondrocytes, we see that researchers can scrutinize disease-specific cellular phenotypes and molecular pathways. So those have a deepening on our understanding of the syndrome's pathogenesis. So iPSCs derived cell models facilitate the exploration of molecular mechanisms, and they underlie Aarskog syndrome pathology, shedding light on dysregulated pathways and potential therapeutic targets. So this paves the way for the development of targeted therapeutics tailored to the unique genetic and molecular profiles of individual patients. So in summary, I know this was a lot. In summary, iPSCS technology holds an immense promise in advancing Aarskog syndrome research, and it offers insights into disease mechanism and the laying foundation for personalized therapeutic approaches to improve patients' outcome. Here we have the reference used on this presentation as well as the written project proposal. So thank you so much for listening. I hope that you understand a bit about what Aarskog syndrome was about and how it functions and how it signals in a biochemical basis. Thank you so much.